Welcome back. We are looking at module number 6, which is on reaction equilibria. We had looked at the conditions for reaction equilibria and then the equilibrium constants, the temperature pressure dependence of the equilibrium constant, and then we worked out a few problems with which we reviewed or we brought back to memory, if you can call it so, whatever has been learnt in the earlier classes starting with higher secondary. Today is the pretty much the last class on module 6. The next class will be a review for the entire course. In this class, let us look at a formalism for handling electrolytes. Why do we need to look at electrolytes at all? This is a bio course, a biological systems course. We are applying thermodynamics, classical thermodynamics to analyze biological systems. Why are we looking at electrolytes at all? Because most biological systems deal with charged species and ionic solutions. Even if you go back to the 3 examples that we considered one after another in the last couple of classes, you would have seen the occurrence of ionic species all over there. There was ATP, the NADH was a charged species. We talked about half reactions, reduction potentials, and so on and so forth. And if you look around and see many reactions that occur in the cell, you would see the occurrence of ionic species in them. Not just that, the cytoplasm itself, the cell envelope and what is inside, apart from the organelles, is the cytoplasm. It is filled with ionic species. If you look at some of the major biomolecules, there are 4 major types of biomolecules, as you can recall: carbohydrates, proteins, lipids, and nucleic acids. And out of these, if you look at lipids and proteins predominantly, and even the other types in some specialized cases, they all have charges on them. For example, lipids, as you know, the lipid bilayer is the one that is essential for the functionality of the cell membrane. And what exactly is a lipid? It is a charged species. It has these tails, the hydrophobic tails, and the hydrophilic head. The hydrophilic head has a charge on it. This is, you would have heard examples of lipids on the membrane, and most of them are charged species, and that kind of a structure is necessary for its function. Similarly, proteins, amino acids that make up the proteins have charged side groups on them, and even some of the other groups could be charged depending on the pH at which they operate. And if you put a sequence of amino acids together, as in the case of a protein, the protein molecule is definitely charged. Nucleic acids are charged, and some carbohydrates are certainly charged. Therefore, you find charges in all these basic constituents of biosystems, the biomolecules, and therefore we certainly need to look at the charges. And if you want even further motivation, the processes involving electrolytes are used to separate and analyze DNA or proteins. We typically run gels in which we separate out the various parts of the DNA, analyze them, various proteins in a protein mixture, analyze them, and so on and so forth. So all these are electrically active processes involving electrolytes, and therefore it is important to know the formalism for handling electrolytes from a thermodynamics viewpoint also. This is a slight extension of what you would find in your textbook. In fact, you will not find electrolytes in Smith, Van Ness and Abbott. Nevertheless, I thought since this is the last class, I will take the liberty of introducing you to something slightly beyond your syllabus. It certainly falls into the scope of the course, and it will be good to know. and if you do not want to use it for your exam, please feel free to ignore it. You have 38 or 39 other lectures for your exam. Let us consider the electrolyte. As you know, electrolyte is any species that is charged, and let us represent the electrolyte as M nu plus A nu minus. Now, M could be the positively charged species, A could be the negatively charged species. Nu plus is the number of positive charges associated with M, nu minus is the number of negative charges associated with A, and together they could be an uncharged species also, not necessarily, but they could be uncharged also. So this, when it is dropped into a solution, for example when it is taken in a solution form, it dissociates as follows to give you nu plus molecules, or if you take one molecule of M nu plus A nu minus, it will dissociate in an aqueous solution, for example to give you nu plus molecules of M Z plus and nu minus molecules of A Z minus by the stoichiometry of the whole thing. This is nothing but material balance, right. So if you extend that to moles, we talked of molecules, and Avogadro number of molecules is a mole, and therefore 1 mole of M nu plus A nu minus gives you nu plus moles of Mz plus, the positively charged species in the electrolyte, and nu minus moles of the negatively charged species in the electrolyte. We will get a little more comfortable with this terminology in a little while, please wait till then. It is good to introduce it in a generic form here. Let us call this equation 651, and as for completeness, nu plus and nu minus are the numbers of positive and negative ions respectively from one molecule of the parent electrolyte. Z plus and Z minus are the number of ionic charges, and of course as you will see soon, this nu plus and Z plus need not always be equal. Similarly, nu minus and Z minus need not always be equal. Nu is the stoichiometric part, and Z is the number of charges associated with each molecule. To understand the terminology a little better, let us work out an example in the same fashion that we have been doing so far. For that, let us consider a relevant system here. The following reaction system is important in the pH maintenance in the media of cultured mammalian cells, bicarbonate system. See, for example, you may have come across mammalian cell culture. Mammalian cells are cultured in very many different vessels. For example, they are passaged in what is called a T flask, a tissue culture flask, which is typically placed inside a carbon dioxide incubator. You have a supply of carbon dioxide to that incubator. Incubator is something that maintains the temperature, humidity conditions, and in this case carbon dioxide is also supplied to the incubator. Why is it supplied? Because that is essential for maintaining the pH of the solution to be in a very narrow range of, let us say, 6.8 to 7.2. Even finer, that is the wide range over which many mammalian cells are even alive. Below 6.8 they are all gone, and above 7.4 they are gone. Typically the bicarbonate system, which involves the equilibrium of a bicarbonate salt in the medium with the carbon dioxide that is supplied in a gas form, is necessary to maintain the pH of the solution. I will show you the reactions, and then I will let you figure out how the pH is maintained on your own. You just need to go and look at the bicarbonate buffer system for mammalian cell culture. You can do a search, you can look at any books on mammalian cell culture. That is one of the first things that will be covered there. The question here is relevant to whatever we are doing here. It is to find nu plus, Z plus, nu minus and Z minus. The reactions are bicarbonate giving you H plus and HCO3 minus, and HCO3 minus giving you H plus and CO3 2 minus. These in fact are the reactions that are that play a role to maintain the pH in the mammalian cell medium in a narrow range. So, because the mammalian cells are going to grow, when they are going to grow they are going to produce lactic acid is going to move from the inside of the cell to outside of the cell into the medium. If there is acidification due to lactic acid, the pH is going to drop. Therefore, the medium should be able to withstand at least a reasonable acidification or the addition of acidic ions, lactic acid H plus ions to the medium, and that is done this way. Similarly, on the other side, if the pH goes up by due to some means, then this medium must be able to handle that. I am not getting into the mechanism of the bicarbonate buffer itself, that I am letting you that I am leaving to you as some additional reading. You can go ahead and do it. As of now, please find nu plus, Z plus, nu minus and Z minus. Take about 10 minutes to do it. Go ahead please. Okay. It is essentially an exercise to get comfortable with the terminology, the stoichiometric coefficients as well as the number of charges that are associated with these molecules. The reactions that we are considering here are H2CO3 going to H plus and HCO3 minus, that is the first reaction. So for that you need to write nu plus Z plus nu minus Z minus, you would have done that, and do the same for the second reaction. So the solution is reasonably straightforward. Just by comparison you can see that nu plus is 1, Z plus is 1, nu minus is 1 and Z minus is minus 1. If you are wondering, excuse me, where this came from, for the first reaction, nu plus is the stoichiometric coefficient of the positively charged species, so that is 1 here, therefore nu plus is 1. There is one charge associated with the positively charged species, therefore Z plus is 1. Nu minus is again 1 here, the stoichiometric coefficient of the negatively charged species, and Z minus, there is one negative charge associated here, therefore Z minus is 1. In contrast, here Z minus is, in other words Z minus is minus 1, because that is the convention that we follow, we add the sign also to the number, therefore Z minus is taken as minus 1 here. In contrast, here Z minus would become minus 2, as we will see in the solution given. For the second reaction, please go ahead and check, you would have already gotten this. Nu plus is 1, Z plus is 1, nu minus is 1, this by the same logic as the first reaction, but Z minus is actually minus 2. Okay. Now the components of the solution, that is what we are going to look at. That the solution, let us say, contains m moles of the electrolyte. You know how we represent our electrolyte, m plus nu plus, a minus nu minus, electrolyte dissolved in N0 moles of the solvent. If this is the situation, this is how we will represent things, electrolytes in our formulations. M moles of an electrolyte dissolved in N0 moles of the solvent. If that is so, then the notation is N0 is the moles of the solvent, that is okay, as given here. M plus is taken to represent the moles of the positive ions Mz plus, M minus is taken to represent the moles of the negative ions MaZ minus, and Mu is the moles of the undissociated electrolyte M nu plus A nu minus. The reaction that we saw earlier, M nu plus A nu minus giving you nu plus M Z plus and nu minus A Z minus, the number of moles of this that exists at equilibrium when the dissociation has taken place is called Mu, and this results from adding m moles of an electrolyte, which would be of course different from Mu, to N0 moles of the solvent. That is the system that we are considering. Now from the stoichiometry as given here, it is quite easy to see that the number of moles of positive ion as represented by M plus is nothing but nu plus, because 1 mole of this results in nu moles of the positive ion, nu plus times M, M is the total number of moles of the electrolyte, minus the moles of the undissociated electrolyte. If Mu is what is remaining here, M minus Mu must have converted to this, and 1 mole of this gives you nu plus moles of this, and therefore M plus equals nu plus into M minus U. Similarly, M minus would be nu minus into M minus Mu, equation 652 and 653. Now let us get to a thermodynamics thing. Let us look at the chemical potentials involved, and equation 215, one of the basic equations, you will remember that, when you see that written here. If you write equation 215 which involves Gibbs free energy, total Gibbs free energy, written for our current system of an electrolytic solution, would be dGt equals, now it is coming back to you, minus St dt plus Vt dP plus mu I, sum over I mu I dNi, and going by our terminology and writing each one of those terms separately, we get mu plus dm plus mu minus dm minus plus mu undissociated mu of the undissociated species times dmu, the differential of the number of moles of the undissociated species, plus of course you have the solvent there too, mu 0 dn 0, this corresponds to the solvent. We will call this equation 654. Now if we impose conditions of constant temperature and pressure, the first two terms are going to go to 0, dt is 0, dP is 0, and if we use equation 652 and 653 to express m plus and m minus, remember we express m plus in terms of the number of moles of the electrolyte added and the number of moles of the undissociated electrolyte, just the two equations before this. Let me write it and it will come back to you. If we do use these two expressions, equation 652 and 653 to express m plus and m minus, we get dGt, the first two terms have gone to 0 already, equals mu plus, which is here, and dm plus is nothing but nu plus into dm minus dmu, quite easy to see this, and similarly you have a nu minus here, and dm minus is nu minus into dm minus dmu plus of course nu u dmu plus nu 0 dn 0, the number of moles of the solvent, and you can rearrange this as dGt. I have essentially collected all terms with the dmu together, nu u minus nu plus mu plus minus nu minus mu minus dmu, from here there is a dmu here, there is a dmu here, and there is a dmu here. I have just collected the coefficients of those and written and collated them in terms of dmu, dm and dn 0, and so the first term was mu u minus nu plus u mu plus minus nu minus mu minus dmu. The second term I have collected in terms of dm, nu plus mu plus plus nu minus mu minus dm plus mu 0 dn 0, equation 655. And now let us use the lines of the argument used to derive the condition for reaction equilibrium. Now what is the, if you recall the lines of argument, it was that we had free energy and the free energy changes with the reaction coordinate or the number of moles, and therefore if you take the partial derivative of that with respect to the reaction coordinate under certain conditions, under certain constant conditions, then since this is the minimum at equilibrium, the slope of this curve, the free energy versus the reaction coordinate or the number of moles, the slope of that curve would be 0. We will use the same lines of argument here. Dou n1 of Gt at constant T and P must be equal to 0. This is the condition for equilibrium, which is carried on from our early courses even. This also formalizes equation 6-7 in this module. But here the variables, m is the number of moles of the electrolyte solute which is dropped into n 0 moles of the solvent, and it results in mu number of moles of undissociated electrolyte. All these three are independent of each other. You could take some number of moles of the electrolyte solute and in some number of moles of the solvent, and depending on the condition some number of moles of the undissociated electrolyte will remain, the rest would have dissociated. In other words, these are not dependent on each other, that is the only point that I am trying to make here. These are independent of each other, whereas if you consider these three variables, mu, the number of moles of undissociated electrolyte, the number of moles of positively charged electrolyte, the number of moles of negatively charged electrolyte, these are related by the equilibrium distribution. At a certain point in the dissociation, it can dissociate only to a certain number of moles of mu plus and a certain number of moles of the positive species and certain number of moles of the negative species, that is governed by equilibrium. Therefore, only one of them can be considered to be independent, whereas the other two will depend on that based on the equilibrium conditions. Therefore, only mu can vary while the equilibrium is being attained for a given number of moles of solute M taken in a certain number of moles of the solvent N0. Once we are clear about these things, it is quite easy to write the functionality dependencies. We can write the condition for equilibrium in an electrolytic solution as dou Gt dou mu mu. We are just taking it with respect to one independent variable, the number of moles of the undissociated electrolyte at constant Tp, M and N0, the number of moles of all other species and the number of moles of the solvent. This is 0, this is the condition for equilibrium, equation 666. And note, if the total differential is considered in 655, dGt equals under constant conditions of temperature and pressure was nothing but mu u minus mu plus minus nu minus mu minus dmu plus nu plus mu plus nu minus mu minus dm plus mu 0 dn0. This, if we write in terms of partial differentials, it is easy to see this is dGt. Therefore, this would be dou Gt dou mu at constant other things. I am not going to mention the constants here. Dou Gt dou mu dmu, dou Gt dou m dm, dou Gt dou n0 dn0. Therefore, dou Gt dou mu at constant Tp m n0 is nothing but this term mu u minus nu plus mu plus minus nu minus mu minus. Let us call this equation 667, and now we know where we are getting at. This is equal to 0, so we are going to equate this to 0, and if we do that we get nu u equals nu plus mu plus plus nu minus mu minus. In other words, the chemical potential of the undissociated species must equal the sum of the chemical potentials of the charged species weighed by its stoichiometric coefficients. Equation 668. Now, in equation 49 we saw a formulation for chemical species that is helpful in solutions of biological relevance. Now, this was for solutions which is different from which makes it convenient and so on and so forth. Similarly, there is another formulation that is used for electrolytes, where the solute concentrations are expressed in terms of molality, L instead of R, molality, which is moles of solute per kilogram of solvent. It is not moles of solute per volume of the solvent, it is moles of solute per unit mass of the solvent rather than molarity. Molarity, of course, you know, is moles of the solute per liter of the solution. whereas here it is moles of the solute per kilogram of the solvent. And if we use this formulation, then we get for the solvent mu naught equals mu naught hash plus RT ln gamma naught x naught, and of course gamma naught tends to 1 as x naught tends to 1, and for the solute we have mu I equals mu I triangle, if you want to call it that. This is based on molality, moles of the solute by the mass of the solvent, plus RT ln gamma I mi, this is in molal units, moles of the solute by the mass of the solvent, and the activity coefficient gamma I tends to 1 as mi, the molality, tends to 0. Let us call this equation 669. Therefore, the chemical potential for the solutes in an electrolytic solution, let us say it contains only one electrolyte to begin with, we will deal only with one electrolyte in this course. This is anyway an extension to you, and if it gets more complicated, you can get into a research mode and find out how it is handled. It is already been done in papers and so on. This can be defined as mu plus equals mu plus triangle plus RT ln gamma plus m plus. Let us call this equation 670, and I am just writing the chemical potential for each of these species separately. Mu plus, mu minus equals mu minus triangle plus RT ln gamma minus m minus, equation 671, and mu u, the chemical potential of the undissociated species, is nothing but mu u triangle plus RT ln gamma u mu, equation 672, where these mu triangles, the standard values, are functions of temperature and pressure, the same way as mu hash was, and of course gamma I tends to 1 as mi tends to 0 in all these cases, as to be in line with the definition that we have given earlier. Now, if we substitute equation 670 and 671 in 668, what do I mean by that? 670 and 671 in 668 was this. This is the condition for ionic equilibrium. Mu of the undissociated species is sum over nu I mu I, or in this case nu plus plus nu minus mu minus. So, if we substitute it there, we will do that in a little while. We get nu u equals nu plus, I have replaced the mu plus with mu plus triangle plus RT ln gamma plus m plus, and mu minus by mu minus triangle plus RT ln gamma minus m minus. Now, let us look at the right hand side alone and just combine the terms. I am just combining terms appropriately to make a point. Mu plus plus nu minus mu minus plus RT ln gamma plus m plus to the power of nu plus, there is a ln here, there is a nu plus here, therefore, this, if it is taken inside the ln, it becomes a power, therefore ln of gamma plus m plus nu plus RT ln gamma minus m minus raised to the power of nu minus, just a rearrangement, which would result in mu plus triangle plus nu minus mu minus triangle plus, there are two logarithmic terms here added together, and if you put them together, the addition of logs is log of the products, and therefore plus RT ln gamma plus raised to the power of nu plus gamma minus raised to the power of nu minus m plus raised to the power of nu plus m minus raised to the power of nu minus, equation 673. If the total number of positive and negative ions is nu, that is what it means, and if we define a mean ion activity coefficient as gamma plus minus and a mean ionic molality as m plus minus. What do I mean by that? There are these terms here. I am just trying to simplify these terms. There are two terms, gamma plus to the power of nu plus, gamma minus to the power of nu minus, and similarly m plus to the power of nu plus, m minus to the power of nu minus. I am going to replace this with some mean value, I am going to replace this with some other mean value, that is what I am doing here. So gamma plus minus to the power of some nu equals gamma plus nu plus times gamma minus nu minus, and therefore gamma plus minus is defined as gamma plus nu plus gamma minus nu minus to the whole raised to the power of 1 by nu. Let us call that 675, and on the same lines m plus minus is m plus raised to the power of nu plus, m minus raised to the power of nu minus, and the whole raised to the power of 1 by nu, the lines of this 676, and therefore using the mean values in terms of the above definitions, that is what it is, we can write the previous equation as nu u equals mu plus raised to the power of mu plus triangle, the standard value, plus mu minus triangle plus nu RT ln of gamma plus minus m plus minus. These are the mean values that we have defined here. Let us call this equation 677. Now let us look at the equilibrium constant for the electrolytic reactions. It is nice to know of an equilibrium constant for the dissociation process and related to the chemical potential. To do that, into the condition of equilibrium 677, which is the mu u equals mu plus nu minus, if you substitute the expression for mu, whatever we had written in terms of the other variables, we get minus RT ln of gamma plus minus raised to the power nu m plus minus raised to the power nu divided by gamma u mu equals mu plus triangle plus mu minus triangle minus mu u triangle. Just do this exercise on your own and see whether you get it. It is a one step process, you must be able to get this. Equation 678, and now it must be clear to you, following the same strategy as for non-ionic reactions, we can see what the equilibrium constant is going to be defined as. We are trying to get a minus RT ln k term here, therefore k is defined as this one inside the brackets, gamma plus minus raised to the power nu m plus minus raised to the power nu divided by gamma u mu. Let us call this equation 679, and since the values of the terms inside the square brackets, which is this or their constituents m plus and m minus instead of m plus minus, are sometimes available in tables. So there is a way by which you can a priori say what the equilibrium constant of an electrolytic reaction would be, and that is what makes this powerful. So prediction of the equilibrium constant is possible if these values are available. Now we are going to take a slight detour to establish useful temperature dependencies for estimating the relevant thermodynamic variables here, the equilibrium constant and so on and so forth. For that we are going to write the second part of equation 669. What is 669? Let us go back and show that to you in yep. This was the formulation for the chemical potential of the solvent and solute in terms of the special way for electrolytes. Solvent remains the same, whereas solute was mu I equals mu I triangle plus RT ln gamma I Mi, where Mi is in molal units. We are going to look at this particular equation now. We can write the second part of the equation of 669 that we just saw as mu I triangle by T equals mu I by T minus R ln gamma I minus R ln Mi. Now mu I was mu I triangle plus R T ln gamma I Mi. So I just split it up as this and divided by the temperature, therefore these two T terms have gone. Now if this equation is differentiated with respect to temperature at constant pressure and composition, which means Mi is a constant and therefore the derivative with respect to temperature is going to be 0. So if we take if we differentiate this with respect to temperature, then we have dou T of mu I triangle by T equals dou mu I by T dou T minus R dou ln gamma I dou T here, and this term of course has gone to 0. Let us call this equation 680, and using 515, which will become, we have seen this so many times, so it will come back to you. Dou T of mu I triangle by T is nothing but minus HIT hash by T squared, the partial molar entropy, molar enthalpy by T squared. This we had shown by an elaborate procedure in equation 515, minus of course the remaining term R dou ln gamma I dou T, equation 681. And now let us consider the limiting composition case, that is Mi tending to 0 when gamma I tends to 0, and according to the definition given in 669, under such conditions equation 681 can be written as dou mu I triangle by T dou T equals minus Hi at infinite dilution, okay, Mi tending to 0, infinite dilution, Hi infinity hash in the partial molar value at infinite dilution divided by T squared, equation 682, where this is actually called the partial molar enthalpy of component I, which is the electrolyte at infinite dilution. Now if we do a similar exercise with the first part of that equation divided by T and take the derivative with respect to temperature again and so on, this is the first part of equation 669, that would give us dou T of mu naught hash by T equals minus H naught by T squared, 683, where H naught is the enthalpy per mole of the pure solvent. Now if you substitute 683 into this 681, we are trying to substitute one of these values here, a 682, or let us further consider 683 before we even substitute that. Note that mu I triangle is not dependent on composition, therefore dou T of mu I triangle by T does not vary with composition. Therefore follow this argument a little carefully, we have done this argument earlier too. At the limiting value where the molality has gone to 0 and gamma has gone to 1, the same equation 683 is valid. So it does not depend on composition, therefore this equation must be valid even under these limiting conditions of infinite dilution, and therefore under these conditions if we substitute 682 and 681 we get dou ln gamma I dou t equals hi infinity hash minus hi t hash divided by r t squared. We are just substituting this into this, minus hi infinity hash by t squared onto this and getting ln gamma I dou ln gamma I dou t, an expression for that. Dou ln gamma I dou t equals hi infinity hash minus hi t hash by r t squared. We will call this equation 684, and what this gives us is the temperature dependence of the activity coefficient. If we do a similar exercise for substitute for the solvent, the 0 case in the same equation, then we will get dou ln gamma naught dou t, this is for the solvent, equals h naught minus h naught t hash divided by r t squared, and therefore by knowing the right hand side we can get the dependence of the activity coefficient on temperature for an electrolytic system. Equation 685. This is all I have for the electrolytes, and when we meet in the next class we will do a review of whatever we have done in this course.